Okay, so welcome to this next video in which we are discussing angiogenesis in chronic inflammation. Okay, so we've seen now that in chronic inflammation what you're going to get is angiogenic factors, which are vascular endothelial growth factor A and fibroblast growth factor 2. They will come to the vascular endothelial, specifically of the post-capillary venules, and they're going to trigger growth in these endothelial cells, and we are now looking at the pathways by which this is achieved. Okay, so we've seen that both vascular endothelial growth factor A and fibroblast growth factor 2, they have tyrosine kinase receptors, and basically when these tyrosine kinase receptors become activated, you get that autophosphorylation where the dimer basically phosphorylate each other, basically. So receptor 1 phosphorylates the tyrosine residues on receptor 2, and receptor 2 phosphorylates the tyrosine residues on receptor 1. Okay, and then what's going to happen is these phosphorylated tyrosine residues on the intracellular aspect of these receptors are going to trigger all sorts of downstream pathways, 3 to be precise. We are looking at the first one now. So we have seen that a Ras-Gef complex has assembled, a Ras-guanine nucleotide exchange factor complex has assembled, and it's going to activate the G protein Ras. It's going to chop off guanosine diphosphate, and it's going to add on guanosine triphosphate, which takes the Ras into an active state. The Ras GTP, or this on state of Ras, is then going to bind to the kinase enzyme RAF, and that activates the RAF kinase enzyme, and it's now going to phosphorylate the MEK1 enzyme, which stands for MAP kinase, or MAPK/ERK kinase 1. Right, and MEK1 is activated by this phosphorylation, and it then phosphorylates another enzyme. Okay, so here comes the next enzyme, and when you phosphorylate this next enzyme, again it goes from being in the inactive state to now being in an active state. So again, here is a phosphate group stuck onto this kinase enzyme. Okay, and again this is a serine threonine kinase that we're activating. So we're getting this kinase cascade effectively. We activate one kinase so that it can activate another one, and then that one will activate another one, etc. Okay, so this enzyme is a very important enzyme. Okay, so it's what is known as MAP kinase, okay, and this stands in full for mitogen activated protein kinase. So this is the mitogen activated protein kinase. Okay, it can also stand for microtubule activated or associated, that's the correct one, microtubule associated protein kinase. But we'll think of it as the mitogenous activated protein kinase. Okay, now this enzyme, confusingly, is also known as the extracellular signal regulated kinase. Okay, and this is why it can also be referred to as ERK. So extracellular signal regulated kinase. So extracellular signal regulated kinase. So basically, MAPK is short for MAP kinase. Okay, ERK is short for extracellular signal regulated kinase. Okay, and both of these names are still used. Oh, whoops, this is coming off of view. Both of these names are still used for this same, and it's not just one enzyme, that's the thing. It's a whole family of enzymes. So when we talk about MAP kinase, we're really talking about a whole family of enzymes, okay? And again, when we're talking about extracellular signal regulated kinase, we should really say the extracellular signal regulated kinases, because again, it's a whole family. So let's talk about two specific examples of MAP kinases, or ERKs. Okay, so confusingly, they have kind of very different numbers. So for instance, the enzyme ERK1, so the extracellular signal regulated kinase 1 enzyme, is also called MAP kinase 3. Okay, so MAPK3. Okay, and this is one of the enzymes that will be activated by MEK1. Another one that will be activated by MEK1, okay, which we'll put here, is the extracellular regulated kinase 2. Okay, but confusingly, this is now what's known as MAP kinase 1. Okay, so the numbers don't match up, basically. So MAP kinase 1 is not ERK1, it's ERK2, and ERK1 instead is MAP kinase 3. But these two are important examples of the MAP kinase family, or the extracellular signal regulated kinase family, whichever name you want to use. And these two will both be activated by MEK1. Okay, right. And these both go on to phosphorylate certain transcription factors. So when they've been activated, they will go into the nucleus of the endothelial cell, and they will phosphorylate certain transcription factors. So in come these enzymes into the nucleus of the cell. So let's say this is the nucleus of the cell here. Okay, so here's the nucleus of the cell, and I've just realised that I'm now going to have to try and squeeze everything in to this tiny little space now. Okay, so in come these enzymes, ERK1 or MAP kinase 3 and ERK2/MAP kinase 1. And basically what they're now going to do is they are active serine-threonine kinases. So they're going to phosphorylate other proteins on serine and threonine residues. Okay, so two of their targets are the transcription factor ELK1, okay, and also the transcription factor MYC, okay. So we're going to phosphorylate ELK1, and we're also going to phosphorylate MYC. Now both of these transcription factors are initially inactive, but when they get phosphorylated they're going to become active. Now we'll discuss ELK1 first, because it's going to have a less direct pathway than MYC, okay? So we'll start with ELK1. Okay, so ELK1 is what's known as a transcription factor. So let me just explain what a transcription factor is. And this is going to be a very important concept for this topic. So, in all eukaryotic genes, you have a promoter region upstream of the gene, okay? So if this is the gene here, so let's say this portion here is the gene, and I'll highlight it here in a certain colour. So let me get purple for this gene. So this piece of DNA that I've highlighted and boxed like this is the gene. Now in eukaryotic DNA, upstream of all genes you have another portion known as the promoter region, which I'll show here. Okay, so let me highlight this portion in turquoise. And this is not a portion that will actually be used to make the protein. Instead, it is a portion that controls the expression level of the gene, basically. So in turquoise, this is the promoter region. Okay, so how does it control the expression of the gene? Well, in order to actually make the gene, in order to make the protein for which this gene codes, you need the enzyme RNA polymerase to come and bind to the DNA and work its way along the DNA and synthesize a piece of mRNA which has a complementary sequence of organic bases to the sequence on the coding strand of DNA. Okay, so the RNA polymerase has to bind somewhere, and it basically binds to the promoter region. So it will bind to the promoter region, then it will work its way along and create the mRNA for the gene. That mRNA will then go to the, it will go out of the nucleus, and it will be translated by a ribosome and made into the protein. So basically, how much mRNA you produce is going to determine how much protein you produce. Okay, So, if the promoter region has a very high affinity for binding to RNA polymerase, then RNA polymerase will continuously come and bind to this promoter region and will make a huge amount of mRNA for this gene, and therefore you'll get a lot of translation of the protein, so you'll end up with a lot of protein for that gene. Okay? Whereas if the promoter region has a very low affinity for RNA polymerase, then RNA polymerase will only very rarely bind to this promoter region. You won't get that much mRNA synthesized for this gene, and therefore you won't get much protein produced. Now, a transcription factor is something that comes and binds to the promoter region. So here is our transcription factor, and the transcription factor will alter the affinity of the promoter region for binding RNA polymerase. Okay, so you can have transcription factors which will enhance the transcription of the gene, i.e. they will increase the affinity of this promoter region for the RNA polymerase, and therefore increase the amount that the RNA polymerase binds to the promoter region, and therefore increase the amount of mRNA you produce, and therefore the amount of protein you produce. Or you can have repressors, which will decrease the affinity of the promoter region for the RNA polymerase, and therefore you'll get less mRNA being produced, and therefore less protein being produced. So transcription factors, by altering the affinity of the promoter region for RNA polymerase, can alter the expression of the downstream gene. Okay, so ELK1 is going to bind to the promoter region upstream of certain genes and increase the expression of those genes. And one of the genes it's going to increase the expression of is actually another transcription factor, which is known as CFOS. Okay, so I'll put this here, CFOS. And basically CFOS can assemble with another transcription factor known as CJUN. Okay, so here's another transcription factor known as CJUN. And the two of these can assemble together to make a CFOS-CJUN heterodimer. Okay, which I'll try and show in here. Let me show this here. So I'll have to show it by colouring in these two. So let's colour in CFOS in this red colour here. And this can be a CFOS here. And then let's show CJUN in orange. Okay. And the one above the CFOS can be the CJUN. Okay, so you get these CFOS-CJUN heterodimers being formed. So this is a CFOS-CJUN heterodimer. Okay, so it's a dimer because it's got two constituent proteins making it up. It's a heterodimer because the two constituent proteins are different, basically. Hetero means different. Now, CFOS and CJUN will do the same thing. Well, sorry, the CFOS-CJUN heterodimer will do the same thing as MYC, and that is, they're both going to act as transcription factors, and they're going to increase the expression of genes which will make the cell grow. However, we will come back and discuss CFOS-CJUN heterodimers and MYC once we've discussed the two other pathways that the receptor tyrosine kinase are going to start up, basically, because the other two pathways as well are going to lead to transcription factors which will do the same thing as MYC and CFOS-CJUN. Okay, so let's turn our attention to these other pathways now and see which transcription factors they're going to activate. Okay, so we'll continue this discussion in the next video.